Sharing the screen now. Oh, I don't have my headphone on. Just a minute here. Now you'll see me struggle to put this on. Okay, can somebody confirm that you're hearing me? Can you guys hear me?
Yes.
Thank you. I think I'm sharing the screen. Let me check. No, I'm not. There we go. I don't know if we need that. So if you look on the schedule, we're going to be finishing chapter 13, viruses, viroids, and prions today. And we might get started just a little bit, a few slides on chapter 7. I think we will have time just to get started. And then that'll make an easy next week. Next week we will have the chapter 7 on Tuesday, and then on Thursday we will review for the final. On Saturday of next week, the unknown project report is due by 11:59 p.m. Saturday night. This week there are no labs, the same as next week. I will be logging in from 6:30 to 6:45 if anyone has questions. So if you have questions, please log in between 6:30 and 6:45. If you're unable to log in and you have questions, you can always email me. Any questions about any of that? All right, so we talked about COVID-19 last time. We mentioned that its symptoms are pretty much like the flu. The differences being that, of course, there's higher deaths and there's shortness of breath with COVID-19, which are usually not seen with the flu. Although you should note that the flu can kill people. So we do have deaths every year from the flu. So it too can kill people, but it's less likely to kill than COVID-19. And yes, it is true that the majority of the people who die of COVID-19 are people who have underlying health problems. But even among young people who get COVID-19, death can occur. So take precautions about getting COVID-19. And the best thing is to get a vaccine. And I can state that I was reading yesterday that the experts are thinking that the vaccine looks like it's going to last a year. They don't know for sure how long the vaccine will last, but it definitely lasts more than 6 months. Just a note about COVID-19. Let's move on to the topic of viruses and cancer. Some human cancers, between about 10 and 15% of human cancers, are caused by a virus. And when I say cause, I'm going to use that term loosely, that the virus seems to have some role in initiating the cancer. Because even today, we do not exactly know how cancer comes about, but the virus can start the process or encourage the process. Some examples: human papillomavirus is associated with both cervical cancer and skin cancer. Hepatitis B and hepatitis C viruses are associated with liver cancer. And they think that these viruses are getting in these cells and then encouraging the cells to become cancerous. That doesn't happen with all cancer. A recent study, actually it's not so recent anymore, by Tang et al. In 2013 found that breast cancer in human and glioblastoma is not associated with viruses, so that some cancers occur without the patient ever getting a virus that then tips them into getting the cancer. It's just some cancers. And you'll notice that it's a minority of the cancers in humans that are caused by viruses, but some of them. I'm not going to talk about cancer. You can take a look at this. Cancer is uncontrolled growth of cells. Viruses can cause cancer in two main ways. Viruses can have the formation of a provirus where the viral genome integrates into the host cell's DNA, resulting in a permanent change in the host cell, changing the host cell's DNA. If you think about it, with a retrovirus, I gave you that example, that viral genome integrates into the humans' DNA cells, and the integration is not always in the same spot. In fact, it seems to be random, that the virus integrates into a chromosome, and in different cells it can be in different chromosomes. So when that viral genome integrates into the host DNA, it can muck up the host DNA, causing a permanent change or even a mutation in a gene. Imagine retrovirus inserting, what would be a good example of a gene? We'll just say the catalase gene. Of course, that's not going to do too much, but it would knock out the catalase production in that cell. Any question about that? So the formation of a provirus can create a permanent genetic change in the cell, leading to a mutation in that cell. And therefore, viruses that integrate into our DNA can cause cancer. And I didn't mention that, but let me even see if that's in the previous slide. No, it's not. Cancer comes about when the cell generally mutates. At least that's the theory, that a normal cell becomes cancerous after that cell has mutated, and actually more than once. The second way that viruses can cause cancer is that some viruses contain an oncogene, and we call these oncogenic viruses. An oncogene is a mutated form of a normal cellular gene involved in cellular growth. So for our cells to divide and grow, we have genes that control that, control the cell dividing and control the cell growing, and then make the, like for example, the child grow, where they get more cells. Well, an oncogene is a mutated form of one of these genes involved in cellular growth. And these mutated oncogenes can help transform a normal cell into a cancerous cell, because the normal cell that has the normal genes for growing suddenly becomes infected with a virus that carries an oncogene. Now this cell no longer multiplies and divides at the normal rate. It multiplies and divides at an elevated rate, and it can transfer the oncogene into that cell, causing that cell to become cancerous. Any question about any of that? So an oncogene, when it gets into one of our cells, it's generally bad because that cell will start growing. And when the cell grows beyond the normal, growth rate, we call that a cancer. Simply growth in one area is cancer. Anyways, any question about any of that? All right, I didn't hear any questions, so we're moving on. Let's talk about latent and persistent viral infections. We talked about lytic animal viruses and budding animal viruses. Now let's talk about latent animal viruses. Some viruses do grow, and usually they're budding viruses, but they could also be lytic viruses. And then for reasons of their own, the virus stops multiplying and enters the latent life cycle. When a virus enters the latent life cycle, the virus remains asymptomatic in the host cell and in the patient. And it can be asymptomatic for long periods of time. When the virus enters the latent life cycle, the virus does not cause disease. It's like the virus just goes dormant. It's not growing, it's not multiplying, it's not causing disease, it's entered dormancy, and it's not really doing much. It's just staying there. And then suddenly the virus becomes active again. It leaves the latent life cycle and enters the multiplication life cycle, which is usually the budding life cycle, but it could also be the lytic life cycle. Two examples of viruses that enter a latent life cycle are the virus that causes cold sores, herpes simplex virus type 1. When you have a cold sore, like on your lip, the virus is multiplying. It is not in the latent life cycle, it's in the budding life cycle. And then the cold sore goes away, and it may go away for years, or at least a year, and then you come down with another cold, and then you get another cold sore. And that's when the virus leaves the latent life cycle and then enters the budding life cycle, causing the cold sore. A better example of latent virus is a child comes down with chickenpox. The virus is multiplying when the child has signs and symptoms of the chickenpox. The virus then enters the latent life cycle, and it can be latent for years or decades. And then the child is an elderly adult, and the virus leaves the latent life cycle and multiplies and causes shingles. If you don't know, the virus that causes chickenpox, the very first time you get it, you have signs and symptoms of chickenpox. If ever you get that virus entering the multiplication cycle again, it no longer causes chickenpox. It will always cause shingles, and every time the virus leaves latency. Most people get chickenpox, and then that's it. They never get shingles. But about 15% of patients who have the chickenpox can get shingles, and usually it's when they're elderly. It's not always, and usually they only get shingles one time. But occasionally you have people who can get shingles multiple times in their life. I knew someone who got it about every year. So almost every year, I think it was in the wintertime, she came down with a shingles infection. The trigger for these diseases, meaning the trigger for the virus leaving latency, is usually a manifestation of some stressor. For cold sores, the stress is usually when a patient comes down with a cold, and then the person gets the cold sore. But it doesn't have to be that. I'm not sure what the stressor is with shingles. It may be that the patient's immune system becomes weak, and when they become elderly, their immune system usually deteriorates, and that might be the stressor for shingles. Another life cycle that animal cells have is the persistent viral infection. So animal virus can be lytic, it can be budding, it can be the latent life cycle, and it can be the persistent viral life cycle. And in reality, there is at least one more, but we're not going to talk about it. So let's talk a little bit about persistent viral infections. These are different viral infections. When a patient comes down with a persistent viral infection, they get a disease, but the disease processes occur over a long period of time. Usually the disease starts mild, and then the patient continues having the disease, and it worsens over a long period of time. Detectable infectious viruses build up, so the patient initially is only shedding a little bit of virus, and then with time more virus, and over a long period of time the patient just continues to shed more and more virus. The virus is never cleared by the host immune system in a persistent viral infection. A persistent viral infection may be fatal. I think all of you are familiar with AIDS. The patient gets sicker and sicker, the viral load gets higher and higher, the patient sheds more and more HIV virus, and then the patient may die. Now, AIDS used to be fatal in almost all cases. Today, with medication, we can keep the patients alive for a long period of time. And we still consider that a persistent viral infection because the patient has the disease for a long period of time. Another example, which I'm sure most of you aren't familiar with, is subacute sclerosing panencephalitis, which is caused by the measles virus, but it doesn't cause the measles, it causes panencephalitis, and it gets worse over time. Not all persistent viral infections are fatal. Many of them are tolerated. Like about half of people in the world get a cytomegalovirus, persistent viral infection, and they're continuously shedding cytomegalovirus, and that's abbreviated CMV. Varicella zoster, where patients have the chickenpox virus for long periods of time, and I'm talking about persistent. This is a patient who constantly has shingles for a long period of time. My mother had that, where when she got shingles, it looked like it was going to be, she was going to have shingles for the rest of her life. And in fact, that was the case, because she died having shingles. When she got it, the shingles never went away. There are a few other herpes viruses that cause persistent viral infections. All of these are herpes virus. HIV is not. It's a retrovirus. When we look at the different types of viral infection, An acute infection is one where the disease happens very rapidly. The patient comes from shedding no virus to shedding a very high number of virus, and then the disease lasts for a short period of time. The patient gets better, and they shed no viruses. A classic example of that is the human cold. The disease happens rapidly. When the patient is sick, they're shedding large numbers of viruses, but the disease lasts for a short period, and then the patient gets better, and they're no longer shedding the virus. This is an example also of the flu, but most people don't consider that a short infection. But when we talk about viral infections, the colds and the flus are both short and both follow an acute infection, where initially the patient is well, shedding no virus, then they're shedding a large number when they're sick, and then very rapidly, relatively speaking, they get better and they're shedding no viruses. A latent virus infection is one where initially the patient has an acute infection and then they have a latent infection later. When a child normally gets the chickenpox, they get sick, they're shedding large numbers of viruses, they get better over a short period of time, they're shedding no viruses because the virus has gone latent, and then maybe years later or decades later, they're shedding more viruses when they have shingles, and you'll note they're not shedding as much as when they had the chickenpox. And most patients do get better, and then they stop shedding the virus. Anyways, that's a latent infection. A persistent infection is when the patient gets sick, they're starting to shed low numbers, the disease is very mild, and over time the disease gets worse, the viral load gets worse, and then you'll note that over a long period of time, suddenly the virus has jumped up in the number of viruses that the patient is shedding, the disease becomes much worse over time, and they're shedding a large number compared to earlier. And you see how the graph is broken? That's because sometimes in a persistent viral infection, the patient dies. Any question about any of that? All right, I'm hearing no questions, so let's move on. Prions. Prions are not a virus. They are a proteinaceous infectious particle. And this is the theory, meaning they've never quite proven it because we've never taken protein, from an infected patient who has symptoms of a prion disease and then given them to another patient and then watch that patient come down with the prion disease. But it's believed that prions are caused by an infectious protein. They can be inherited and transmissible by ingestion, transplant, and surgical instruments, meaning if surgery instruments get the infective protein and they then use the surgical instrument on another patient and it still has the infectious protein on it, you can transmit it that way. Ingestion, when you ingest a prion. Transplant, this has happened occasionally when a patient gets an organ transplant and the organ has prion proteins in it. The patient will then come down with prion disease. Inherited is that there's really, as far as I know, only one gene that when it is mutated, meaning it's not the normal gene, it's mutated in a patient, and they may inherit that mutated gene, they may get prion disease. And usually what happens is the gene is expressed later on in life, so that when a baby is born, the gene does not cause the prion disease, and the baby grows up to be an adult, and the adult reproduces, and then later on in life the adult expresses the prion gene and they come down with prion disease. Any question about that? And that is how it can be inherited, and that is the gene is not really expressed until after the patient reproduces. So the inherited gene is transmitted. And this is when the prion disease runs in families. The families have a gene which is transmitted from generation to generation, but it doesn't cause the prion disease until later in life. Any question about any of that? All right. Well, prions are proteins that appear to cause spongiform encephalopathies. These are vacuoles in the brain that you can see in humans and other animals. In animals, most of the cases are caused by bovine spongiform encephalopathy, which is more commonly called mad cow disease. And partly we can say that that is because of the mad cow disease that happened in England, and that's why we saw so many cases of bovine spongiform encephalopathy. But it can happen in other animals as well as in people, and we've seen it in sheep for centuries, and in sheep it's called scabies, which is a disease of sheep that sheep ranchers have been living with in Europe for centuries. In cervids, it's also called cervid chronic wasting disease. All of these diseases are prion diseases, where an infectious protein is transmitted in one way from one animal or from one, person or animal to a person. And usually, like in scabies, it's believed that the sheep is consuming the protein. I don't know anything about cervid chronic wasting disease. In bovine spongiform encephalopathy, or mad cow disease, the cows were consuming the infectious protein. In humans, we don't call it any of those names. We call it Creutzfeldt-Jakob disease, and I'm not German, so I could be butchering that pronunciation. Or Guttmann-Straussler-Schneckner syndrome, also another German name, or fatal familial insomnia, and Kuru. Any question about any of those? This one, I'm going to just abbreviate it CJD, or Guttmann-Straussler-Schneckner syndrome, tend to be diseases that run in families, so it's caused by an inherited gene. Kuru was actually caused by people consuming the infectious protein. We'll talk just shortly about that. So prion diseases, remember, are infectious proteinaceous particles. Kuru was a disease seen in the Forb tribe Papua New Guinea. And it was discovered in the early 1900s when Europeans got into close association with the Fore tribes, especially when the anthropologists were studying the tribe. What happened is the patient gets sick of the prion disease, the Kuru. They become unable to walk, the loss of the ability to swallow or chew, drastic weight loss, and eventually ends in death. How it is transmitted was consumption of the prion. In the Fore tribe, it was a funeral practice where people cooked and then ate their dead relatives. So if a member of your family died, in the funeral, you cooked the dead person and then ate them, or at least part of them. And this was part of their funeral practice. And that led to them getting Kuru, because if somebody died of Kuru and then the patient was cooked and then eaten, they came down with Kuru. Cooking does not kill the prion, and we'll talk a little bit about that in a bit. But fortunately, the mothers of the Fore tribe noticed that they and their children were coming down with Kuru. And so they, on their own, in the early 1900s, before the 1950s, stopped eating their dead. But when the men continued eating their dead, and the anthropologists came to the island and they saw that the men of the Fore tribe were coming down with Kuru, and they wrote about it. The missionaries in the early 1900s came in Papua New Guinea, and they thought, oh my gosh, this is a really horrible practice, and they put an end to it. They really got the government to pass laws and said that eating of dead people is against the law, and then they punished people who did it. And that actually put an end to Kuru. So if you're talking about the missionaries harming the culture of the tribe, in this case, in my opinion, and this is just my opinion, the missionaries actually did a good thing for the Fore tribe of Papua New Guinea, because they made it so that they no longer came down with Kuru. And they didn't do it to stop the transmission of Kuru. They did it because they viewed the eating of dead to be, I don't know, irreligious. Any question about any of that? But it is interesting that the mothers of the Fore tribe noticed the association between the eating of the dead and the disease happening where Kuru happened. And they stopped their children from eating the dead, as well as themselves. And they got rid of Kuru among themselves by noticing that association. Any question about any of that? Spongiform encephalopathy is where the brain literally looks like a sponge, and that's from the plaques forming, the prion plaques forming in the brain cells and then causing large vacuoles where a normal brain tissue looks like this. And here we have Kuru, classic CJD here, and then scrapies here. And this may be scrapies of a sheep. This is of a patient, obviously, so is that. Anyways, it makes the brain look sort of like a sponge. Why it's called spongiform encephalopathy. Any question about any of that? All right, let's talk about the prion proteins and how they cause disease. The mechanism of disease is that the prion protein is similar to a normal protein in the cell of a nerve cell. So the PRPC protein is a normal cell surface protein. But the protein that causes prion is abbreviated PRP, what do you call that, superscript SC. And so this protein causes prion disease. This protein performs a normal function in a nerve cell. I'm not sure anyone knows what that normal function is, but this protein is expressed in nerve cells and is needed by the nerve cell. The only difference between the PRPC protein and the PRPSC protein is the folding pattern of the amino acids. And I thought I had a picture of that. I apparently don't have a picture of that anymore. So I'll just tell you that the prion protein and the normal protein contain the same amino acids. The proteins have the same sequence of amino acids. The only difference is the two proteins have a different folding pattern. So the protein has a different shape. How the Scrapes protein, that's what that SC stand for, or the prion protein multiplies, is it comes up to the normal protein and then converts the normal protein into a Scrapes protein, or the prion protein. The cell says, oops, I need more of the normal protein, and then it makes more of the normal protein. The trouble is this cell now has two prion proteins. If either one of them come up to the normal protein, they'll convert it into the prion protein. And more prion protein accumulates in the cell. So more and more prion protein accumulates in the brain cells, forming the plaques in the brain cells. And then we'll see vacuoles, and then the bovine encephalopathy. Eventually, the cell will die, and the patient will get worse and worse, and then the patient will die. Any question about any of that? So this is a really different disease caused by a protein, and the protein simply comes up to the normal protein and converts it into the prion, or disease-causing protein. And then that leads to cell death and eventually the death of the patient. Any question about any of that? All right, I'm not hearing any questions. That's it for prions. So let's talk a little bit about plant viruses and viroids. Plants also have viruses that infect plant cells and cause disease in plants. The plant viruses resemble animal viruses in many ways. They do cause diseases of economically important crops, like plant viral diseases in beans, corn, sugarcane, potatoes. And actually potatoes, tomatoes, and gosh, what is that? Tobacco are caused by tobacco mosaic virus, which can be spread from smokers' cigarettes, and then the patient touches it the cigarette with their fingers, and then they can spread it to the tomato or the tomato plant from just touching the plant without washing their hands after touching the cigarette. Because of the tough cell wall that plant cells, viruses tend to enter the plant cells through wounds or via parasites like a nematode, fungus, or an insect wound. Infected plant cells can then spread the virus. Often the virus gets spread in pollen or seeds, but obviously a nematode or a fungus or an insect can also spread the plant viruses. Any question about plant viruses? Alright, we're not going to talk much about plant viruses because we're not talking about plants. Plants do not cause human disease, and plant viruses do not cause human disease. So this is all I'm going to talk about with plant viruses. Let's talk a little bit about viroids. Viroids are naked RNA molecules that cause disease. You can think of them as a virus, a naked virus, without the protein coat. So it is a piece of RNA that causes disease. Fortunately, the viroids that we know of are all plant pathogens. So the viroids only cause plant disease. There are no known viroids in animal cells. They are infectious RNA molecules, and like among the potato plant, they can spread from something carrying the virus from one plant to another, and then they cause potato spindle tuber disease, where the potato plant grows smaller potatoes than normal. So farmers hate this because their potatoes are smaller than normal. And another thing is that it tends to spread pretty easy. All right, any question about plant viroids? Naked RNA molecules causing plant disease. Here we're looking at an actual electron microscopic image of a plant viroid. And all it is a naked piece of RNA. All right, the last slide in this topic is, in recent years we found new huge viruses, very large viruses that infect amoebas. For some reason, amoebas have these very large viruses. Both megavirus and Pandora virus, which are the largest viruses we know, infect amoeba. Pandora virus is as long as a bacteria. Okay? Its diameter is a little bit shorter than a bacteria, but its length is as long as a bacteria. So this is a huge virus. A megavirus is about 500 nanometers in diameter. A typical virus is only around 100 nanometers or less, like influenza A. All right, any question about viruses, prions, and viroids? If there's no questions, I'm going to end this lesson and start our next lesson. Chapter 7. We'll stop early today. I just want to get started on Chapter 7 and make it easy next week. So Chapter 7 is the topic of controlling microbial growth, usually with agents that disinfect or antisept something. But you could autoclave or boil or simply clean something. And this is a topic that students generally have a pretty good feel for. We've all used disinfectants. We've all used antiseptics. So students generally have a good feel for this lesson, which is one reason to have it last, and that is it'll be a fairly easy lesson for you. The major goals and in rough outline of this lesson are given in this slide. First, know the terms: sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, biocide, and germicide. In addition, be able to discuss the factors that influence microbial death rates, be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells from growing. Fourth, be able to understand the various types of disinfectants and how they work. And lastly, number 5, understand the relative resistance of the major microbial groups to antimicrobial agents, meaning one antimicrobial agent may work better on some groups of microbes because of the characteristic that that microbe has. All right, any question about what we're going to do for Chapter 7? Any questions by anyone at all? You guys are a really quiet group. All right, when we're talking about the rate of microbial death, you should realize that bacterial populations die at a constant logarithmic rate. So when we plot, let me blow this up a little bit, plot the death of cells over the time, and the time follows a normal axis, meaning a normal, what do you call that, scale, but we plot the number of cells on the y-axis. Cells die on a logarithmic plate at a constant rate. Here it's graphed, and it depends on the age and how quickly the cells die. But in this one, they're dying at 1 log decrease for every unit of time, meaning 90% of the population die in 1 unit of time, another 90% later after 2 units of time. If you were to plot that on an arithmetic number of scale, meaning right here, you wouldn't get a constant death rate. The death rate would be like that, okay? And that's why we plot it on a log scale. Another way of looking at that is with this table here, and then as you increase the time, the number of survivors decrease by tenfold each unit of time. So if we have 1 million cells, to decrease it to 1 cell that's alive, you need to expose that cell to the agent for 6 minutes of time, because there's a constant death rate of the cells over time. Any question about that? So if you have a large number of cells and you want to get rid of them and you treat them with an agent, you need to treat them with that agent for an appropriate length of time. And the more cells you have, the longer time you need to expose the cells to that agent. Any question about any of that? I think you all kind of know that, but needed to talk about it. All right, so the rate of microbial death can be influenced by a number of factors. The effectiveness of the treatment depends on the number of microbes that are present at the beginning of the treatment. The more microbes you have, the longer you need to treat the agent or the microbes with the agent. I meant not the agent, the unit you're treating. You need to treat it longer. Environmental influences can affect the rate of microbial death. For example, the presence of organic matter, which is almost always seen in hospital specimens, can influence the effectiveness of the treatment on how quickly they will kill the cells or how many cells they will kill. For example, bleach is a very good antiseptic or disinfectant, but bleach is easily inactivated by organic matter. And so if you have a lot of organic matter around, the bleach will become inactive or inactivated and then will no longer kill the cells. Any question about any of that? All right, not hearing anything. The temperature of the treatment can affect the effectiveness of the microbial death rate. Generally, a higher treatment will increase the cell killing. The presence of biofilms can influence the microbial death rate. Cells that have a biofilm will die less readily than cells without a biofilm. And so if cells have a biofilm, you have to treat them with an agent for a longer period of time. Any question about that? The time of exposure has to be increased depending on the characteristics of the microbe. For example, endospores will require a much longer period of exposure to kill the endospores than cells without endospores. And some endospores will be resistant to that treatment. Like some endospores can resist treatment with bleach. I don't know why some endospores do die with bleach, but some do not. It must depend on the characteristics of the endospore. And then we'll talk about microbial characteristics at the end of this lesson. If cells have certain microbial characteristics, they will be harder to kill. For example, Mycobacterium has mycolic acid in the cell wall. And if you're treating the cells with an agent which dissolves in water, that agent will not get through the mycolic acid of the cell wall, and that agent will not kill the mycobacterium. So the microbial characteristics can also influence the microbial death rate. Any question about any of this? All right, I know that's a lot in this slide, but if you think about it, there are just 4 characteristics that influence the microbial death rate. So that's pretty good. This slide is showing you the different terminology we use for controlling microbial growth. Let me blow this up. So you should know these terms: sterilization, destruction or removal of all forms of microbial life that includes endospores. It does not include prions. I never mentioned that with prions. Prions are very hard to kill. You need to get the temperature up to 1,000 degrees Fahrenheit to kill a prion. And most of us never have exposure to 1,000 degrees Fahrenheit. Our ovens can't get that hot. Obviously, boiling water won't get that hot. The microwave won't get that hot. So, in your home, I would say it's pretty safe to say you cannot destroy a prion. So sterilization in the microbiology lab is usually done by an autoclave, which is similar to a giant pressure cooker, and that is how we sterilize the media in a microbiology lab. Commercial sterilization, you don't really need to know anything other than the term. Sufficient heat treatment to kill endospores, especially of canned food, and that's to kill off things with Clostridium botulinum. It's more important in the food industry. Disinfection is destruction of vegetative pathogens. It generally does not kill endospores. A disinfective agent is something we use on an object, like when we disinfect the toilet. Antisepsis is when we use an agent to destroy vegetative pathogens on living tissue. So an antiseptic is something we use on living tissue, like maybe iodine to make the skin aseptic. Usually antiseptic is less harsh than a disinfectant. But if you use the same agent on an object, we call it a disinfectant. If you use it on living tissue, we call it antiseptic. For example, if you use bleach on a toilet, it's a disinfectant. If you use bleach on a patient, it is an antiseptic. And actually, I don't know if this is still used, but with the flesh-eating bacteria, one of the early treatments they found that was effective at treating that bacteria was to put the, arm or whatever, your leg, in something wrapped with bleach and expose the flesh-eating bacteria to the bleach. And yes, the bleach was harmful on the skin, meaning you'd get a little bit of a burn from the bleach, but the disease was much worse than the bleach, and that is why they use bleach as an antiseptic on those patients. And like I said, I don't know if they've come up with another treatment that was an early treatment they came up with for flesh-eating bacteria. Degerming, that's the removal of microbes from a limited area, such as degerming the area for injecting into that area. So when we're alcohol swabbing a region to inject, we're certainly not sterilizing the region, nor are we making applying an antiseptic to the region. We're degerming the area, just reducing the number of microbes in the region. Any question about that? Sanitization, another treatment used in the food industry to lower microbial counts on eating and drinking utensils, to bring the microbes down to safe health levels. And actually, you do that when you put your dishes in the dishwasher. Okay? You sanitize them by putting them in the dishwasher. All right. Let me go ahead and talk about these terms, and then we'll end for today. Some terminology to control microbial growth. A biocide or a germicide is a substance that kills a microbe. It does not usually kill endospores, but it does kill the parent cell of an endospore, which we call a vegetative cell. A bactericide is an agent that kills bacteria. A fungicide, an agent that kills fungus. A virucide, an agent that inactivates viruses. And all of these would be a biocide or a germicide, but these are specific in for what they're killing. Bacteriostasis is not killing the microbe, but inhibiting the growth of microbes. You remove the bacteriostatic agent, growth might resume. And a simple example of bacteriostasis is when you put something in the refrigerator. Let me think of an example. A human pathogen which will not grow in the refrigerator, Staphylococcus aureus, but it will survive in the refrigerator. But if you put it in the refrigerator, you will inhibit its growth. It will no longer grow. But then you remove it and bring it back to room temperature, the Staphylococcus aureus will continue its growth. And that would be bacteriostasis. There are agents which are bacteriostatic. And for the life of me, I can't think of an example except for some antibiotics are bacteriostatic, where they don't kill the bacteria, they stop the bacteria's growing. I can't think of an agent that we apply on an object or a tissue that's bacteriostatic, but I'm sure that there are some agents that exist. All right, any questions about any of this? If not, I'll see you next time. If you have a question for the lab, remember to log in. Why isn't this going? Remember to log in at 6:30 to 6:45. I'll be there to answer your questions. Okay?